Neisseria gonorrhoeae, the pathogen causing gonorrhea, is an increasingly resistant and difficult bacteria to treat on the global scale. The mechanisms of resistance that it employs are well described, yet we still continue to struggle with its antibiotic resistance. The implication of a completely antibiotic-resistant case of gonorrhea is becoming all the more possible. The most commonly described resistance mechanism found in Neisseria gonorrhoeae is the evasion of the antibiotic. The target of most antibiotics in Neisseria gonorrhoeae is the protein PBP2, coded for in the PEN-A gene. Therefore, mutations, as shown to the left, to this gene cause resistance to these antibiotics, and these mutations are fairly common in highly conserved bacteria. The mutations confer resistance by causing structural changes in PBP2, consequently leading to the inability of the antibiotics to bind to it, thus causing resistance. Another very common mechanism of resistance is the overexpression of efflux pumps. As shown in the picture, efflux pumps allow for bacteria to pump out substances that pass through the cell membrane, and the efflux pump that confers resistance in Neisseria gonorrhoeae is the MTR-CDE efflux pump. In wild-type strains, the repressor MTRR binds to the DNA, leading to normal expression of the efflux pumps. However, mutations in the DNA coding for the repressor significantly decrease the amount of repressor in the cell, in turn significantly increasing the expression of the efflux pumps, thus allowing Neisseria gonorrhoeae to pump out enough antibiotic to survive. Overexpressed efflux pumps and mutations of the antibiotic target simultaneously in the same cell are not uncommonly found and are characterized by high-level resistance to many antibiotics. Neisseria gonorrhoeae has this ability to be very resistant, but many cases are actually treatable. The recommended treatment for gonorrhea is a dual therapy of ceftriaxone and azithromycin, but in 2018, there was a recurring infection in Canada that indicated a failure of the recommended treatment. What if this highly resistant strain spreads globally, as many resistant strains already have? Well, a team of researchers have created a chimeric protein fused to an immunoglobulin M, shown to the left, that seems to circumvent this antibiotic resistance mechanisms. This new treatment binds to the bacterial cell surface to enhance complement-dependent killing of Neisseria gonorrhoeae. Bacteriophages are also being tested to treat bacterial infections due to their high specificity to kill only a single bacterium. In summary, the resistance ability of Neisseria gonorrhoeae is evolutionarily formidable. The mechanisms that it employs are simple, yet have proven to be highly effective in the survival of the bacteria and portray the danger we face without efficacious treatment. This is a scary predicament, but there is hope in the treatments we currently have and the ones that are being made. The recommended treatment continues to be very useful, and the use of chimeric proteins has already proven to be efficient in clearing gonorrheal infections. In order to stay ahead of Neisseria gonorrhoeae and its resistance, more research must be conducted on chimeric proteins as well as the potential of bacteriophages.